Hello everyone. Today we will discuss about tests for carbohydrates. First we will see Molish test. This is a general test to detect the presence of carbohydrates. In positive result, violet or purple ring appears at the junction between sugar and acid. It means carbohydrate is present in the sample. No reaction means carbohydrate is absent. Next one is iodine test. This test is used to distinguish mono or disaccharides from polysaccharides. Here no reaction means simpler carbohydrates. For example, if the sample contains lactose, glucose, fructose, sucrose, then there will be no reaction. If blue or black color complex is there, it confirms starch. Then red means glycogen, brown means dextrin. So iodine test will be positive for polysaccharides like starch, glycogen, dextrin, etc. After that, next test is known as Barfoed's test. Barfoed's test is a chemical test used to detect the presence of monosaccharides. Here disaccharides may also react, but the reaction will be much slower than monosaccharides. This test detects the presence of monosaccharides, which detect reducing monosaccharides in the presence of disaccharides. So this Barfoed's test distinguishes reducing monosaccharides from disaccharides. The presence of red precipitate detects the presence of reducing monosaccharide in the sample. If the color appears within 2 to 3 minutes, the sample contains reducing monosaccharides like glucose or fructose. If the color appears later than 10 minutes, it means the sample contains reducing disaccharides. That is Barfoed's test. Next one is Sullivan's test. This test distinguishes between aldose and ketose sugars. This test is a confirmatory test for ketohexose, that is fructose. If a blue-green product to peach product, that means the sample contains an aldose. If a cherry-red product is there, that means the unknown sugar is a ketose. For example, fructose. That is Sullivan's test. Next one is Biochemistry test, which is used to detect the presence of pentoses. Blue or green product indicates the presence of pentoses and muddy brown to grey product means the presence of hexoses. That is Biochemistry test. Next one is Benedict's test, which is used to detect the presence of reducing sugars. The difference between Barfoed and Benedict's test is Barfoed's test is used to distinguish between mono and disaccharides, whereas Benedict's test is used to determine reducing or non-reducing sugars. This Benedict test is usually done after Barfoed's test. If a brick red precipitate means reducing sugars are present and no reaction means the solution contains a non-reducing sugar. That is Benedict's test. Next one is Ozone test, which is used to detect reducing sugars. These are the examples of crystals which are formed in Ozone test. For example, for glucose and fructose, needle-shaped crystals. Galactose, thorny ball-shaped crystals. For maltose, sunflower or star-shaped crystals. For lactose, cotton ball or powder puff-shaped crystals. So this is Ozone test. So this is about the test for carbohydrates. This is today's topic. Thank you for watching.